All right, welcome to the last lecture for chapter 13. So in the previous videos this week, we talked about X-linkage and what that means, but I just want to kind of review X-linkage and give you some examples of where it shows up in nature. So one of the examples, and we've done some problems with this already, or you're about to do some problems with this, is red-green color blindness. Now, one of the things I'm going to have you guys do this week during your lab portion of the week is to actually go take the color blindness test. Red-green color blindness means that you can't differentiate between shades of red and green. So you see them all generally as one shade of, like as a darker or lighter type of one color. So they can't see all those different colors. You'll kind of get a better idea for what colors are hard to see when you take the colorblind test, because if you have full color vision, you can kind of see how they're using colors to sort of hide the pictures in that colorblind test. Another one is Duchenne's muscular dystrophy. So muscular dystrophy, this is a special type that occurs more often due to X-linkage and then hemophilia. What we notice about an X-linked trait is that it's going to always show up more often in boys. We talked a little bit about why this is, but it's because that gene is carried on the X chromosome. So let's say that you get a mutated gene that says you should have color blindness, okay? And so I'm going to use this little sort of bar on the top of the X to show a colorblind gene. Now, if you're a girl and you have, or a female and you have one normal chromosome and one with the colorblind mutation on it, you will still have normal vision. So this person would still have normal vision. Okay. For a girl to be colorblind, she would have to get a mutated X from her dad and one mutated X from her mom. So her mom would either have to be a carrier of colorblindness or would have to be colorblind, and her father would have to be colorblind. So this would be a colorblind female, and I'm just going to write CB and then female. So this would be a colorblind female's genotype. Now let's look at what has to happen for males. Males only have to they have one X chromosome and then the Y. So now if a male has a normal X, then obviously he would be normal, right? We'll just put N-O-R. Normal. But as soon as a male inherits one X with the colorblind marker on it, he now is colorblind. Okay, so there's no such thing as a male being a carrier for color blindness. This female, we would call her a carrier, meaning she doesn't exhibit the trait, but she has a gene, she carries a gene for that trait. So she's a carrier, but she is not, she doesn't actually have color blindness. For males, you can't be a carrier of color blindness. You can only either have it or not, because it's what we call X-linked. So that would be an example of X-linkage. Black and orange hair colors in cats are also due to genes on the X chromosome. So what that means is that in cats, you should either only have a male that is black or a male that is orange. If you have a male cat and that cat has more than one hair color, it means that your cat is actually a Klinefelter cat. And what that means is that your cat has extra chromosomes. So if you have a male cat that has more than one color, it's actually this genotype. It has an extra X chromosome. Because if we have males here, an X with a black gene and a Y would mean that this would be a black cat. Follow me? And an X with an orange gene, because that's the only chromosome the male has, would mean that it is a orange cat. Okay. Now, in order to get a cat that is like modeled, or what we would call tortoise shell, you would have to have that be a female cat normally. And that female cat, because she has two X chromosomes, would have one black and one orange. Now, in certain cells, the black chromosome would be expressed, and so those cells are going to create black hair. In other cells, the orange chromosome is going to be expressed, and so we'll get orange hair, which is how you get these cats that have these different colors of hair. I'm gonna just go forward because I think I have a picture of this. Okay, yeah, so this, you know, this would be a black cat. This would be what would happen with the male. So just full on black. Okay, and then we have this would be considered full on orange. And then this is what we would call tortoiseshell. Ah, so good at writing with my mouse. Okay, so this would be what we would call a tortoiseshell, and then this is what you might call calico. Both calico and tortoiseshell should be female-only patterns of inheritance, because they require 2 X chromosomes. So this patch, for example, would be expressing the black X chromosome, and this patch here is expressing the orange X chromosome. Now you might say, well, why is there white at all then on this cat? And this is actually cool. This is a 3rd gene. The 3rd gene is preventing either color from being expressed, so we get white fur. So it is possible to have like a black and white male cat, but this color patterning or this tortoiseshell patterning should not be happening in male cats unless they are Klinefelter, which I just explained. All right, linkage on other chromosomes. For this one, I'm pretty much just going to, if you need to pause it to get this note, go ahead, but I'm just gonna go ahead to this picture. So on other chromosomes, Mendel said that all genes should be inherited independently. You guys remember when we talked about independent assortment? Unfortunately, Mendel got really lucky. He wasn't 100% right. So what we learn is that when genes are together on chromosomes, so each of these letters is gonna signify a gene. So we have gene A, gene B, gene C, gene D, and gene E. Now, because crossing over occurs, okay, what that means is that let's say these chromosomes are actually going to cross over. They're going to swap here and here, okay? So this A and B is going to be swapped for this A and B, okay? What that means is crossing over is way more likely to occur where A and B are almost always going to go together, right? Because there's a lot less chance that, let's just get rid of this. There's a lot less chance that we would divide the chromosome. Let's see, so some place between like here and here. So A and E could go separately, right? Because that leaves all of this region of the chromosome. We could cut really anywhere in here. So we could cut here, cut here, cut here, cut here. And so A and E are not as likely to be inherited together, right? Because there's basically 4 different places where we can cut this and have crossing over occur that would result in A and E being inherited separately, okay? However, let's go back and look at A and B, okay, 2 that are right next to each other. So how likely is it that when crossing over randomly occurs, it's going to happen at exactly this place, okay? We don't have a variety of different opportunities for these to happen. So basically we get this rule, and that is that the closer, so physically closer, so the less actual space that's between 2 genes, the more likely they are to be linked or inherited together. So what that means is if we had to like kind of rank these, A and B would be the most linked genes, okay? And then if we had to go to the next level, A and C would be the next linked genes. And then D would be slightly less linked and E would be even less linked, okay? So we can get linkage on other chromosomes, and all linkage means is that the 2 genes are very close together, okay? So A and B might be linked, D and E might be linked, C and D might be linked, but any genes that are close together are going to be linked. Now we're just going to spend a couple of minutes talking about a few disorders that can happen with inheriting chromosomes. And the first and main one that I want to talk about is non-disjunction. So what non-disjunction means is that during mitosis, when the cell was dividing, the chromosomes did not go equally to the poles, right? So if we have a cell that is dividing, right, during anaphase. Let's say that this cell had 2 chromosomes. We should get 2 going up and 2 going down. Non-disjunction would be, instead of getting 2 going up, we get 1 chromosome that basically stays together, and then only get 1 chromosome down here. So one of our cells gets more chromosomes than the other. And this is called non-disjunction. So non-disjunction is when the chromosomes do this. They don't divide evenly, okay? Now, a lot of times in non-disjunction, that's going to be fatal in humans, because if you lose 1 chromosome entirely, a lot of times that embryo isn't even going to continue to develop in a way that it could be a viable pregnancy. So that's one of the major things there. However, there are certain elements of non-disjunction that are survivable. So the first one would probably be the mutation that causes Down syndrome, which is trisomy 21. So what that means is, instead of having 2 copies of 21, they have trisomy. So they have 3 copies, or an extra copy of chromosome 21. Turner syndrome is where you don't get a second sex chromosome. We talked about these during sex determination, but it's a female, biologically female, with only 1 X chromosome. Oftentimes people with Turner syndrome are infertile. Klinefelter syndrome, I was just talking about this with cats. Klinefelter syndrome means you have an extra X chromosome, and so you are biologically male, or the organism is biologically male, but it has an extra X. These can all be diagnosed by karyotyping, which is the lab procedure that I had you guys go over for your lab last week, where we actually take a picture of the chromosomes and organize them in that way. Only Klinefelter cats, only Klinefelter cats can be calical or tortoiseshell male. So we already talked about that. So I'll just kind of skip over it for now. So these are some things that can happen with non-disjunction, just meaning the chromosomes didn't divide evenly. So we have either a whole piece, a whole chromosome, or a piece of a chromosome that is not where it belongs. Polygenic inheritance is something that happens a lot in humans. We're not going to talk too much about this because it gets complicated really quickly, but anytime in humans where you see traits that appear to blend and have like an infinite number of possibilities. So you could be any height, have any skin color, any eye color. And what I mean by any is like not that you can have pink or purple skin, but that there can be an infinite number of variations in shade, right? Like going from darker to lighter. It would be hard to find 2 people that have exactly the same skin tone, right? So same thing with hair color, right? The exact same hair color would be kind of difficult. Eye color. Everybody has these slight variations, and what that tells us is that it's not just alleles on a gene, it's more than 1 allele that are determining these kinds of things. All right, that is it for our last lecture. That wraps up chapter 13. Make sure that you also watch the lectures this week where I went over certain problems from the genetics lessons, because those ones are going to teach you how to actually do all of your homework. So go ahead and take a look at those. And as always, send me an email or hop on Zoom if you have questions. Bye.